This video is showing the in vitro induction of human dental pulp stem cells toward pancreatic lineages.
Stem cells became an alternative treatment in many diseases, including diabetes type 1, which caused by losing of beta cells. Transplantation of insulin-producing cells will be the new promising method for controlling blood glucose lifelong.
In this video, two approaches for generating insulin-producing cells, including the integrative and non-integrative induction protocol, are described. And in this part, the integrative protocol for inducing insulin-producing cells from human dental pulp stem cell is explained. For the induction protocol, is about for mimicking the natural pancreatic developmental process to get the mature and the functional insulin-producing cells.
For generating insulin-producing cells from mesenchymal stem cells like dental pulp stem cells, the multi-step induction protocol is employed. Mesenchymal stem cells are induced toward definitive endoderm, pancreatic endocrine, and then pancreatic beta cells or insulin-producing cells, respectively. To induce the cells, three-step induction approach is used as a backbone protocol. The integrative protocol employed the overexpression of the essential pancreatic transcription factor, PDX1, by human dental pulp stem cells. Then, the PDX1 overexpressed cells will be further induced using three-step differentiation protocol. The pancreatic differentiation potential by the integrative protocol will be compared with the non-integrative protocol as illustrated in